Yeah, thank you for joining us. This is to introduce the newest system that's available within the Centre for Advanced Histology and Microscopy here at Peter Mac. Just in case you don't know me, my name's Paul McMillan. I'm the manager of CAM here at Peter Mac, and we've got two representatives from Nikon on the line as well. So if there's any tricky questions, I will hand over to Dr. Kevin Grant, who's an application specialist here in Victoria for Nikon Health Australia. And he's the one who's going to be doing the presentation today. And you'll also see that Dennis McPhail has just turned on his camera as well. So Dennis is our local service engineer for the Nikon systems as well. So before I pass over to Kevin, I should probably just do a brief introduction. So what I'd like to do before we start is just acknowledge the traditional owners on the land on which we meet, which for myself is the Wurundjeri people of the Kulin Nation, and pay my respects to their elders, past, present, and emerging. And with that, Kevin, I will pass over to you to go through the presentation.
Thanks, Paul. A lovely introduction. And hello to everybody. Yes, as Paul mentioned, my name's Kevin Grant, and I'm the application specialist for Victoria for Nikon Healthcare. I'm kind of excited to be talking to you today about this system and what Paul and the team have managed to, or have installed, and Dennis as well, have installed at the Peter Mac. Really is a pretty fantastic tool with some really pretty unique capabilities. And so what I wanted to do today was just to give you a really quick overview of what that technology is, a sneak peek of how we're able to maximize the imaging outputs that the instrument can provide. But more importantly, it was actually to give you some examples of samples that people have imaged and characterized using this system elsewhere and throughout the world. And so the idea was really, as I say, just to give you a background on the technology, but really to try and ignite some sparks in your minds as to where this system could potentially be used. So without further ado, I will see if I can move to the next slide. Oops, page up. Okay, so what I'm going to do, I'm going to introduce myself and Dennis. Then I'm going to introduce the SoRa spinning disk system that we have operational at the Peter Mac. And then I'm going to use some examples of real samples to outline some of the key capabilities of the system. And then finally, and probably most importantly from your perspective, is have a discussion about how you can get onto the instrument to test some of your samples and to test some of your ideas out for yourselves. So as Paul mentioned, both Dennis and I are on the call here. We thought it would be useful to say hello to you all because both of us are, PhD level trained scientists. And we have, although we work for Nikon Australia, we have a really strong general interest in science and the development of science in Victoria. And both Dennis and I also have worked in that field nationally and in my sense globally as well. So we are just as interested to understand, what you guys are interested in and to talk through ideas, discuss potential applications or potential projects with you as much as anybody else is. So I thought, as I say, it would just be nice for me to say hello, and I'll hand you over to Dennis right now so he can introduce himself as well. Dennis, over to you.
Good morning, all. Yeah, just a quick hello, and hopefully I will see you in the labs over the next few years. So good luck with SoRa. If there's any problems at all, feel free to contact me directly or go through Paul. Paul and Meta would be the best options, but feel free to contact me if you need to discuss anything. So back to Kevin.
Thanks, Dennis. Short and sweet. Awesome. Okay, so that's the personnel part of it. Let's introduce the really cool part of it. Not that we're not cool, it's very cool. So the CAM has, as Paul mentioned, has got a brand new super resolution microscope system. So we've introduced you to Dennis and I, who are the local Victoria representatives for Nikon Australia. What Paul is introducing you to is the local Victoria-based super resolution microscope. Capability-wise, we believe this is the only one of these in the country. And this is an image of Paul's system that I actually took when I was down imaging on it yesterday. It's a super easy instrument to use. Even I can use it, which is a great thing to say. And if I can just give you a very quick rundown of what we're looking at with pulse system. So you will probably recognize in the middle part here an inverted microscope. So this is a Nikon inverted microscope. Attached to the side of that, we have the confocal system over here on the left. And then this is a magnifying changer in the middle. I'll go through each of these components in due course to explain why they're there and what they do. And quite critically on pulse system, we also have up here a thermal controlled chamber with CO2 control as well. So for all of your live cell, live sample imaging projects, this is an essential component. So all of this works cohesively as a really powerful system for really high resolution, really high speed imaging of live samples, really. If we break down what we've got here, as I mentioned, there's an inverted microscope, and there's also this bit of wizardry on the left-hand side, which does the confocal and the super resolution imaging. So in the next couple of slides, what I'd like to do is just to talk to you a little bit about what goes on inside this. This isn't intended in any way to be a comprehensive overview of the system. It just helps, I think, set the scene to help you understand, why it's such a special system installed there. So we're going to talk a little bit about the spinning disc system. So it's called the CSU-W1 SoRa. So SoRa has rather contrivedly derived its name from super resolution via optical reassignment. I think more importantly, SoRa means sky in Japanese. I think they wanted to use that name, and so they manufactured it's an acronym to make it fit, but it sits. So we've got the CSU-W1 SoRa system. This is a spinning disc microscope. So it's different from many other microscopes there, in that it contains within it these microarrays of microscopic pinholes. These are arranged in spirals on that disc, and that disc spins incredibly fast as you're imaging. And what that does is essentially scan thousands of micro beams of light on your sample to build up that image. Because you're using these micro beams of light, it's actually the light budget hitting the sample is incredibly low. And so it really eliminates or reduces significantly the photo bleaching of samples. So it's really useful in that sense for imaging live samples and things like that. So yeah, we'll talk about that in a little bit more. So what does the CSU-W1 SoRa actually mean? The CSU-W1 itself is a spinning disc confocal microscope. As I just mentioned, they're very good at measuring not just beam sensitive, but light sensitive samples. So lots of living samples and things like that can be really safely imaged by this CSU-W1 spinning disc system. Implication of a traditional spinning disc system is that it's basically limited in its resolution capabilities by the laws of diffraction. They can't really beat that. So what the Yokogawa team have managed to develop and what we've implemented into this system here is what they call the SoRa version of this CSU-W system. So this uses a little bit of optical physics, I guess, to increase the resolution capabilities of the system significantly, and significantly to the point that they are greater than, or the resolution capabilities of it exceed the resolution limit as dictated generally by the laws of physics, although we can't break the laws of physics. So it moves the capabilities of the system into the super resolution microscopy field, into the realm of super resolution microscopy. And I want to just spend the next couple of slides explaining to you how the system is able to do that. So bear with me. So what I've done here is just very, very simply explained an optical system on a microscope. So we have the light in, which comes through the emission pinhole. It goes through the objective to the sample, then back through the objective through the excitation pinhole, and it gets collected. So although I've shown the objectives on two sides here, these could be the same objective. The light bounces through, it comes back again. Just for simplicity, I've tried to do this. I hope it works. In the perfect case, what happens is that the light in comes in through an infinitesimally small pinhole. So if it's exceptionally small, there can be very little spread of light through the sample, and the light out at the excitation pinhole is pretty much everything that was taken into the inside pinhole, the emission pinhole, sorry. So this is the perfect scenario. Unfortunately, with the perfect scenario, because this is infinitesimally small, the light budget available to image your sample is also infinitesimally small. So this is not a practical reality in the real world. So what we tend to do, or what happens, is that you have an emission pinhole of a finite size. In this case, we've used a 50 micron emission pinhole. Now, what this means is that although most of the light passes through directly, as it would do in an infinitesimally small pinhole, there is the potential for some of the light that gets emitted from the perimeters or outside of the true optical plane of the emission pinhole to follow a path through the sample that means it deviates slightly from that optical plane. And this can mean, this is very exaggerated here, but this can mean that although much of the light is perfectly focused and collected, some of the light is not. And this can lead to some out of focus light or light loss in the sample, in the collection of the image, which is shown here. So the way that the SoRa system gets around this is quite simply, it's easy for me to say quite simply because I'm not an optical engineer, but I'm sure that in the background this is an awful lot more complicated than I've managed to illustrate it here. They've inserted a micro lens before the light comes out of the sample at the emission excitation pinhole again. And what this in effect does is collects all of this slightly out of focus light and refocuses it very close to optical perfection of the sample. Adding this final micro lens, what this means is, as I say, it gathers more light and it actually improves the XY resolution by approximately 1.4 times with just the standard pinhole. So that means that it's, we've got lots of light coming into the sample and we get more of it coming out. So you're able to increase the signal to noise, it improves the contrast, and it makes it easier to quantify dim and small structures within. So as I mentioned, this is what it looks like in real life. The SoRa part is over here on the left. After that very brief introduction to the physics of the system, what does that actually mean? Why is that useful to you in the lab, I guess? And quite simply, there are three sort of main benefits, I guess. The first of those is that because this system acquires super resolution images optically, there are no special requirements to collecting data from your sample. So you don't have to use additional diffraction gratings or scanning mirrors or any other hardware to collect the images. And you also don't need to pair your samples in any particular way. So all of your standard samples, tagged samples and the like will work perfectly well. Because there's no additional wizardry employed in creating the super resolution images, it makes it really, really fast. And this was a little movie of, I think, neurons shooting across the screen. But because everything is happening optically and live, it means you actually see what is happening at super resolution live on your screen. So incredibly powerful. And the third aspect of the system that is very, very powerful, I believe, is that not only have you got resolution and super resolution capabilities, but you've also got confocal capabilities. And what this means when you couple both of them together is that you have the capability to image deep within samples that are intrinsic to the confocal system, coupled with the super resolution capabilities of the SoRa system. So an incredibly powerful imaging technology. The confocal system can be used alone, or it can be used coupled with the super resolution. So incredibly powerful, incredibly flexible for switching between the different types of imaging parameters should you want to. So I mentioned that the SoRa data, due to the microlensing, improves the imaging capabilities, the resolution of the system relative to a wide field or a confocal system. There is an additional step that can also be added to the system to help improve the resolution even further, and that's by incorporating some deconvolution techniques to the resulting. So incredibly powerful technology with some really incredibly powerful imaging capabilities behind it. This is really just a recap of what I mentioned just a second ago about having the capabilities of having, or it's basically two imaging systems within one. And because it's got two spinning discs, you've got confocal, and then with the flick of a button, you can have super resolution. So within the software, you can flick between confocal and super resolution imaging, and it really is as simple as that. The user interface of the system, there will be optical configurations set up in that. There are optical configurations set up in that. And really, it's just a matter of clicking a button and choosing which of these disk systems you want to use, super resolution or confocal. Oh, let's come back again. Okay. I think that's all I'm going to talk about too much with the spinning disk system. As part of this system, there is also what we call the magnification changer, a very technical name for something that does exactly that. Within this magnification changer, we've got the 1 times magnification for standard confocal imaging, and there's a 2.8 and a 4 times positions within this for use for the 60 times and the 100 times objective. So incredibly powerful in terms of collecting high resolution data. And once again, this magnification changer is all controlled within the software interface on the PC, so the Nikon Elements software interface. So it's motorized and it's a click of a button. So increasing from 1 to 100 times and greater is really just a matter of selecting buttons on the user interface. So if we can wrap up what we've spoken about so far, what does this actually mean? So the potential of the system with the SoRa and with additional deconvolution techniques is to achieve about 120 nanometers lateral resolution. What the SoRa system itself can achieve, so over here on the right, is an improvement of 1.4 times the lateral resolution of a standard confocal system. And what that does is provides us with the capability of imaging using a super resolution microscope. It's incredibly well tailored towards live cell imaging or live sample imaging of different types. This is because the light path itself allows for very gentle imaging, and that very gentle imaging also allows for very, very long duration experiments or characterization of living samples. And I'll show you some of those in a couple of seconds. The second part of it is, so it's incredibly gentle, but it's also incredibly fast. So you see what you see live on the screen, incredibly powerful. In fact, it says that. So it's super easy to use. The system itself is really easy to use. And you can flick between the different modes, confocal or SoRa. As I say, the super resolutions, you actually watch them live on the screen, which is really quite incredibly powerful. As I mentioned previously, there's no special dyes or sample preparation. So everything you would normally do for a confocal imaging, you just bring the samples to the SoRa and you get, improve the resolution capabilities of it, but there's nothing special needed done. And once I mentioned it's suitable, or as I mentioned, it's suitable for really thick samples because of the confocal capabilities. So it says here, including thick samples, 10s to hundreds of microns thick. I was measuring samples yesterday, tissues of brain and intestine yesterday that were 550 microns thick and doing Z-stacks through that. So they were cleared, but we got beautiful images of those. I haven't been able to import them into this presentation, unfortunately. You have to trust me on that. But yeah, so, certainly hundreds of microns thick are perfectly capable, are perfectly achievable in this system. So given that, what sort of samples and what sort of applications, what sort of researchers are using this system right now? So the first thing that I did, excuse me one second, was do a brief literature survey. I apologize slightly for this hastily thrown together slide, but I done a literature search for basically spinning discs or spinning disc publications. Kind of not surprisingly, I guess, this is it's always interesting when you see this. This is something that manufacturers tend to put on their website. They show an increasing number of publications per year. Well, of course there are, because people are publishing, they're going up. But anyway, the point is that within the last sort of 8 years, I guess, but even the last 4 years, the number of publications have really taken off. It really has become a technique that is really proving itself to be incredibly powerful, in generating really high quality data. I also pulled out, some of the publications that these papers sit in. And, we've got Nature there, we've got Nature Communications. So it's really high impact research that's getting published. When we scan the papers and we have a look at the types of applications that people are doing, there's lots of live cell imaging, and there's lots of calcium spikes. Zebrafish are imaged quite a lot. There's multiple organ and whole organism live imaging. And we'll go into some of these in a little bit more. Whole brain images of rodents and other primates, lots of time-lapse stuff. We'll have a look at that. As I was doing, my digging into how many systems there were and how many publications there were coming out of it, I came across this, which I thought was quite interesting and possibly worthy of sharing. So not only are Earth scientists, people on Earth, scientists on Earth using the SoRa to generate research, there's also now a SoRa on the International Space Station. And I thought this was pretty cool, so worthy of sharing. So as you can see here, it was launched in Japan in around May 2020, so just about 2 years ago. It took a few days to get to the ISS, whereby the robotic arms of the ISS grabbed it, took it into the ISS, and commissioned it. So there is actually a SoRa working as we speak, live on the ISS. And what it's doing there, which I think is really quite fantastic, is it's looking at how cells proliferate, how they live, how they move, and how they work in a gravity-free environment. So of course they've got a very unique, I guess, laboratory there to do that sort of thing. I just thought it was pretty cool that there was a SoRa there investigating something as fundamental as that. Quite cool. So back on Earth, what are people using these things for? So look, multiple things. And what I've done is I can't remember if there are maybe 5 or 6 examples here. The first thing that you can do with a SoRa quite comfortably is measure the behavior or the response of things as a function of time. So what we've got here are some HeLa cells. Sorry, my screen's over here on the right, so I can see it bigger. We've got some HeLa cells, as you can see, imaged at 2 wavelengths using the 100 times lens. And this is just one slice, so this is a single, single depth image, but collected for 60 seconds at 3-second interval. What we have here is the response of that cell over that time. Similarly, on the right, we've got a bit of heart muscle, imaged at a single wavelength, but with a slightly different, it's got a bit of depth to it this time instead, so 2 microns depth. So we're looking through the muscle of a mouse here as a function. This is a much better example, I think, or I personally think it's a much prettier example of a Z-stack. That's why I kind of flew over the mouse muscle one, not that it's less important, but this is a 60 micron volume image, Z-stack, of an embryonic stem cell colony. So this is imaged live, as you can see there. A couple of wavelengths, as before, imaged with a 60 times lens. So incredibly powerful, incredibly beautiful images. This is, a similar example, I guess. The mouse embryos were injected, I guess, and confocal multi-layer images were collected over a period of 24 hours, imaging at 10-minute intervals. So, what can we see? As you can see, dimensions are 100 microns deep. They were collected at 1 micron intervals every 10 minutes. So there's a lot of images being collected here, a lot of data. And I think the key points to take out of this are the resolution capabilities of the system. This is the 60 times objective. You can see an awful lot of detail in there. I didn't talk about the crosstalk of the spinning disk. It's a function of the design, but essentially what that means is it minimizes the amount of noise in the system, so it improves the signal to noise of the system, of the sample you're looking at. As I mentioned previously a couple of times now, because of the low phototoxicity of the spinning disk system, it allowed us, or it allowed not us, allowed researchers to collect this long time-lapse image of an early stage embryo. And what we have here on the top right-hand side is that image frozen in time. So on the left, we have the time aspect of it. On the right, we just have the 3D single time snap of that. And I think that the most I mean, it's all pretty amazing, but I think the really astounding part of this is that these bioclasts were then transferred to a recipient mouse that gave birth subsequently to healthy pups. So these embryos actually became mice. They lived. They were perfectly formed and perfectly functional when they were born. So I think as evidence that the spinning disc system is safe to use on live samples, I think that's incredibly powerful. This is very similar to the sample I was looking at yesterday. Not quite as thick as the one I was looking at yesterday, but in essence, what we're looking at here are long duration volume imagings of live tissue. This is a slice of a mouse brain, mouse embryo, sorry, a brain slice of a mouse embryo, collected again with the 60 times objective. 50 microns deep, so not especially deep, but for 2 hours. So an incredibly long and statistically useful imaging experiment. Again, we can image for 2 hours because there's no photo bleaching in the sample. And I mentioned previously as well that depth within the sample is really a problem for the system with the confocal. Zebrafish are very commonly imaged both at CAM and elsewhere on the SoRa system. And the next couple of slides, I've got, I think, 2 slides or so that show just really a kind of sequential characterization of zebrafish using the system. So they start off, I'm going to say rather routine. I don't mean to be flippant, but this is a 20 times dry objective of a zebrafish. And then we've got a slightly more zoomed in version, so 60 times water version of it. So this is a Z stack, about 50 microns deep, as you can see. What this can then lead to, I guess, is live imaging of the sample. So what we have here, I guess, is that what we have here is the twin color imaging of a zebrafish, actually throughout a time period. So 200 times at 1 second interval. So here's 200 seconds of a zebrafish's life captured live on a spinning disc system. This sort of science really captures the imagination of the general public. So this is something that was hosted on YouTube, and as of last night, I checked, 761,000 hits. Or the number of hits has gone absolutely mad, and it's really quite incredible. So what we're looking at here is calcium imaging of a zebrafish brain. You can see the neural activity of the brain. You can read yourself here the imaging characteristics of it, but ultimately what these scientists have done is they believe they stimulated the baby zebrafish by showing it some food, and they believe that the calcium imaging that you're seeing happening is the response of that baby zebrafish to seeing some food. So what we're looking at here right now is, in their eyes, the zebrafish being stimulated by the presence of some. I mean, clearly this requires a really high speed, wide field of view imaging technique, and I think, as you can see here, it's perfectly achievable using the SoRa system. Cool. So, look, in summary, a little bit of a recap, I think, of what we've talked about. I've lost the confocal capabilities from this slide here, but really what I wanted to get across to you today was, give you some examples of what this system is capable of doing. And the key things that it's capable of are the low phototoxicity imaging and really high speed imaging. So what, the number of applications out there, the number of samples that could be characterized by that it's massive. And I think that with the number of publications I showed you, we really are just scratching the surface. So it'd be really interesting to hear from you potential applications or ideas that you have that you think might be applicable to measuring or characterizing on the spinning disc. Excuse me. Yeah, and look, as I say, it's super easy to use. the super resolution images can be seen live, so there's no, you don't have to wait, you don't have to do any post-processing, you see it there, you see it live. Sample preparation is very easy, and as I mentioned before, big samples are a problem. And you have the capability instantaneously of seeing samples and resolution greater than that of the fractional. So I think that the next thing, I guess, is to encourage you all, to reach out to Paul and the staff at the CAM, and then, have a discussion about your samples, about potential uses of the instrument. And of course, Dennis and I are here to facilitate or to help that in any way that we can as well. Dennis and I, of course, have got access to the global Nikon community. I've shown you, I can't remember how many samples there are, maybe 10 different samples here. Given the number of systems we have in other areas, there aren't, this is unique in Australia, this system. But there are researchers using it elsewhere, and we'd be more than happy to facilitate any research potential or ideas that you have, as I'm sure will Paul and Meta as well. So I think with that, if you want to get in touch with Paul, Meta, or myself or Dennis, please feel free to do so. And with that, I think I'll hand over to Paul.
Great, thanks very much, Kevin. So if anyone has any questions, feel free to either stick it in the chat, or if you want to put your hand up, I can unmute you if you want to ask a question directly, either for Kevin or myself. Pause for questions. As Kevin mentioned, if anyone wants to come and test the system out with some samples, then we're open to letting people do that. Hopefully in the next few days, Peter Mac will open up a little bit more for external visitors. So we can even get you in if you're not from Peter Mac, but obviously it'd be nice to see the Peter Mac people on the system, first of all.
I think, Paul, just one thing that you mentioned in the flyer, and I forgot to mention towards the end of my talk. You mentioned the software and the ease of use of the software. I think that really is something, setting up the experiments and collecting data on the system is so incredibly easy. And I think that has to be seen to be believed. So I guess I would encourage people to, to give it a try, rather than me try and explain it by slides or to show anything else. The collecting data on the system is super easy.
Yeah, we've got one question come through. What's the biggest size sample depth that you could image?
Depth is always a very interesting one, because it depends on the scattering, I guess, and how well cleared the sample is. Yeah, it's really difficult.
What's the physical stage limit depth?
Oh gosh,
Dennis might know that one.
Yeah, I'm not sure. He might not. He might not. We'll get back to you on that one. I actually don't know that off the top of my head. I apologize.
Yeah, as Kevin says, the deeper you go, the more scattering you're going to get. So at some point, although the micro lenses on the disc will help you, at some point your image quality will degrade as you go deeper into samples. But yeah, we have zebrafish on there at the moment that are getting imaged pretty well through depth.
I think the response would be give it a try.
Yeah.
Yeah.
So get in contact. So if you don't know either mine or Meta's email, you can just email cam@petermac. What laser wavelengths can the system accommodate? So we've got 6 lasers on this system. So your standard 405. Let me just see if I've got that written down exactly what we've got on. Yeah, so it'll be in the order of 405, 445, 488, 514, 545, 640. Yeah. So you're kind of standard 6 channels with filters to match. So for DAPI, FITSI, TRITSI, Sci5, YFP, CFP.
Well done.
I brought it up because I figured someone might ask. There's something in the Q&A. Oh, that's the second one. Okay, if there's no further questions, then yeah, feel free to reach out to cam@petermac.org. if you want to try the system out. Otherwise, we'll give you back 20 minutes of your time. And yeah, thank you, Kevin, for giving the presentation and the overview.